Now we are going to talk about lab diagnosis of gram-negative bacteria. The first one is Brucella. Now, Brucella is a genus. That's the genus, okay? Under this genus, there are many species. We are not going to mention the names of the species, but we are going to deal with Brucella as a genus. Now, this bacteria usually lives in animals, okay? So basically, it lives in cattle, like, for example, cows. Also lives in sheep and goats. So basically, it lives in livestock. Okay, so how is human infected? So basically, human is infected by, for example, drinking infected milk that is not pasteurized properly. So pasteurization kills Brucella, and also infected he's infected by contaminated meat that is not cooked properly. The third route or the third way is in slaughterhouses. Okay, slaughterhouses. In slaughterhouses, there is if there is a direct contact with the tissues of the infected cattle. So basically, the brucella initially in the infection, it's present in the blood. This phase is called bacteremic brucellosis. So it's present in the blood, also in the bone marrow. So initially it's present in the blood and in the bone marrow. Okay, this phase is usually characterized by fever. Okay, and in this phase, you can take a sample from the blood or from the bone marrow, but usually they found that the bone marrow gives a higher yield of brucella. So it's easier to detect brucella if you take a bone marrow specimen. The second phase, usually the brucella is an intracellular pathogen. Okay, so it doesn't like to stay in the blood for long, so it moves to tissues. So the second phase is the brucella will move to the tissues of certain organs, usually the liver, the spleen, the bone marrow. Okay, and this is called localized brucellosis. Okay, so in this case, I can take a specimen from the infected organ. For example, you can the doctor will notice, for example, an enlargement in the spleen or enlargement of in the liver due to the inflammation, due to presence of the bacteria in these organs. So he can take a sample from the liver or from the spleen, okay, or even from the bone marrow. So it lives in the bone marrow both in the bacteremic brucellosis phase and in the localized brucellosis phase. Now the specimen, you will cultivate the specimen on culture media usually containing blood, because this is a fastidious organism. Okay, so brucella is fastidious, it needs blood. Okay, fastidious organism is a microorganism needing special nutrients for its growth. Okay, so fastidious organism usually it needs blood in the culture media. Now, if you remember, we if you remember in Neisseria meningitidis and Neisseria gonorrhea, in their lab diagnosis. Now, if you remember in Neisseria meningitidis, we said Neisseria meningitidis can grow on blood and chocolate, but Neisseria gonorrhea can only grow on chocolate agar, not blood agar. Okay. But in this case, in Brucella, it can grow on both. There's no problem. There's a medium also called Brucella agar and another medium called serum dextrose agar. Okay, as you can see, also serum. Okay, it comes from blood. Brucella also contains blood. So all these media contain blood. Now, after cultivation, Now we move to serological tests, okay? But before moving to serological tests, we need to understand some immunological basis, okay? Um, the shapes of antibodies, you took this in the first term, but just a quick revision. We're going to deal with IgG and IgM, okay? So now IgG is monomeric, IgM is pentameric. IgM has 10 binding sites, as you can see. This is 1, this is 2, and if you multiply these by 5, 1, 2, 3, 4, so they are totally 5. So there you have 10 binding sites. So that's why it binds to the antigen more. So it binds to more antigens than the IgG. That's why it has a higher agglutination power, while IgG it just has 2 binding sites. Okay. Concerning the affinity to the antigen, IgG is better actually. Okay, so IgM binds to more antigens, but IgG binds to the antigen more efficiently. It means that the affinity to the antigen is higher. Okay, and this is important. Now there is something called anti-human immunoglobulin. Okay, they call it anti-IgG. Okay, this is an antibody for the antibody. Okay, so how do they get this antibody? Basically, they use the IgG from the human. They get the IgG from the human. They extract it from the blood, and then inject it to animals. Okay, and the immune system of the animal will generate an antibody for this IgG antibody. So we end up with an anti-human immunoglobulin antibody, or called anti-IgG antibody. Okay, so this is anti-IgG. So they isolate this anti-IgG and use it. Okay, we will understand the importance of this anti-IgG right now. So anti-IgG, it binds to the IgG intuitively, but doesn't bind to IgM. We need to also understand the titers of IgM and IgG versus time. Okay, so we will start first by drawing the IgM antibodies. So the IgM antibodies are produced initially in the infection, usually after the first week of the infection. So they are produced after the first week, and then they move and they peak at the 12th week after 3 months, and then their titer decreases. Okay, so this is the IgM. Now, when it comes to the IgG antibodies, they are formed starting from the 3rd week, and they peak at the 8th week, and then they continue. Their titer does not decrease. Okay, so this is the IgG. The titer of IgG antibody does not decrease even if the infection is treated. Even if the brucella goes away, these antibodies are like protective antibodies. They stay in the blood and help prevent future infections by the brucella. Now let's start with the first serological test. It's called standard tube agglutination test. Agglutination, it means what? It means basically a reaction between antigen and antibody. So antigen-antibody interaction, that's agglutination. Okay, this test detects total antibody titer. So it detects IgM antibodies to the brucella and detects also IgG antibodies to the brucella, using a standard antigen. Okay, so what is a standard antigen? So basically, they use something called smooth brucella. Now, let's understand that word by word. They call it standardized heat-killed phenolized smooth brucella antigens. So what does this mean? So we have smooth brucella and rough brucella. So what does it mean basically? Let's see. As you can see, so as we can see here, this is a smooth phenotype. This is the smooth brucella membrane. Okay, so brucella is a gram-negative bacterium. I will remind you that it has something called outer membrane. Okay, on the outer membrane there is lipopolysaccharide. Okay, this is the lipid part. It's called lipid A. Those red chains, red zigzag chains. Okay, and you have something called core oligosaccharide, those blue circles. And in the smooth phenotype, there is a green chain called O antigens attached to the core oligosaccharide. Okay, but in the rough brucella, this chain does not exist. This red chain does not exist. Okay, they found that the smooth brucella antigens, okay, when they use the smooth brucella to get the antigen they will use in the standard agglutination test, they found that this smooth brucella gives them better results than the rough phenotype. Okay, let's return here. So we understood what does smooth brucella antigens means. Now standardized. So what does it mean standardized? Standardized, it means the antigens have same concentration. Okay, they have the same concentration. They are standard. Okay, the same concentration, and the concentration is known. Heat killed, of course I need to heat kill the brucella because I just need the antigens. I don't need the bacteria itself. I don't need the living bacteria. I need just the antigens. Phenolized, now phenol is a preservative. Okay, so how does this test work? Now first I will get a serum sample. So I have a serum sample in from the patient, serum sample. Okay, now I will dilute this serum sample. Okay, I will dilute it 20 folds. Okay, so I get a dilution 1/20. So the concentration of the antibodies in the serum sample after 20-fold dilution is 1/20 of the initial concentration in the serum. Okay, then I will make 2-fold serial dilution. So the dilution factors is doubled, it becomes 40. So that's 40 dilution factor. Okay, and 2 folds again, it becomes 1/80. 2 folds again, 1/60, until I reach 1/640. Okay, now I have varying concentrations of the antibody from the serum. Now I will add the antigen. Okay, so I will add a fixed volume of antigen to each of these test tubes, and then I will incubate. Okay, so you will add the antigen to each of these tested tube, a standard amount, a fixed volume of the antigen. I will add them. Okay, and then I will incubate. Okay, so I will leave them for a certain time at 37 degrees, and after incubation I will observe for agglutination. Okay, now I will see strong agglutination in the low dilution of the antibodies. Okay, and as I move forward, as I increase the dilution, I will see the agglutination decreasing. Okay, decreasing till I reach a test tube in which the agglutination is barely visible, after which there is no agglutination. So this test tube in which the agglutination is barely visible, I will use it to determine the antibody titer. So the antibody titer is basically the reciprocal of the dilution of the test tube showing barely visible agglutination. So the antibody titer here is 160. Okay? So basically, if the antibody titer exceeds 160, we will consider this an active infection. Okay? In this case, if it's on the borderline, in this case it's 160, so I need to repeat the test. I need to repeat the test after a week or two to determine if the titer will stay at 160 or if it will decrease or will increase. So an antibody titer above 160 is an active infection. So what does it mean, active infection? Well, active infection can mean an acute or chronic. Well, I can't tell. Acute or chronic, and I can't tell. Why I can't tell? Because basically the antigen binds to both IgM, which is characteristic of the acute phase, and binds also to the IgG, which is characteristic to the chronic phase. Okay, so now let's look at the graph here. If I take the serum sample at this time point, where only IgM is present in the serum, the agglutination between the IgM, the agglutination I will see is due to binding of IgM to the antigen. Now, if I take the serum sample at this time point, where the IgG antibody is beginning to form, the agglutination will be due to IgM and IgG. Actually, the agglutination, the visible agglutination of IgG is not evident unless at very high titers of IgG antibodies. Okay, so here there is agglutination due to IgG, but it's not visible. Now let's move to this time point. The IgG titer increased a little bit. At this time point, okay, the IgG antibody titer is not high enough that it will cause visible agglutination, but however, it's sufficiently high that it will cause blocking of IgM binding to the antigen. Because if you remember, we said that the affinity of IgG to the antigen is higher than the IgM. Okay, so at this time point, I will face a problem in the test. There is a blocking effect here. Here, IgG antibodies cause blocking effect of antibodies, of IgM antibodies. Blocking of IgM. At this time point, there is visible agglutination, visible IgM agglutination, but invisible IgG agglutination. Okay, now let's see the blocking effect of IgM. What is the result of this blocking effect? The result is basically that none of these test tubes will show any agglutination. Okay, because the IgG antibody titer at this time point is not sufficiently high to cause a visible agglutination. So what will the lab worker say at this in this circumstance? He will say that the patient is free of brucella because no agglutination is visible. Okay, actually, if you look at the medical history, you find a medical history saying that, for example, this patient is working in a slaughterhouse. Okay, this patient has fever. Okay, this patient has ingested contaminated milk recently, or infected meat, or whatever. Okay, so there is a medical history, and the patient is showing symptoms. Okay, so in this case, we need to prove this blocking phenomena. So how to prove this? We need to make another test. Okay, but before making this test, before explaining this test, I need to clarify something for you. So this is what happens in the blocking phenomenon. Now you have the IgG antibodies, Okay, they bind to the antigen, the Brucella antigen. As you can see here, this is the Brucella, smooth Brucella antigen. Okay, but they are not showing any agglutination. Okay, so no agglutination, because we said that agglutination due to IgG only visible, because we said that visible agglutination due to IgG binding to the antigen only occurs at very high titers of IgG antibodies. Okay, so what we are going to do in the next test is we are going to add an anti-IgG antibody. Okay, this is anti-IgG. This is the anti-human globulin we talked about in the table, if you remember. This is the anti-IgG. Now, what this anti-IgG will do is that it will make a bridge between adjacent IgG antibodies. Okay, the light chain of the anti-IgG will bind basically to the heavy chain, of the IgG antibody. So it will cause bridging between adjacent IgG antibodies, and thus it will cause agglutination. Okay? So after adding the anti-IgG antibodies to these test tubes that initially showed no agglutination, now agglutination occurs. So now I know that there were blocking IgG antibodies. Okay, so I will deduce that this infection is a chronic infection. So I will deduce that the anti-IgG antibodies bound to the IgG antibodies. So I am sure that there are IgG antibodies, which is indicative of chronic infection of Brucella. So this is called modified Combs test. Okay, so let's go through modified Combs test. We use anti-human globulin to detect blocking IgG antibodies. It's used if agglutination test is negative and the patient shows a strong evidence of Brucella infection. As we have said, it can be medical history or it can be mild symptoms of brucellosis. During the acute phase, let's go through this. During the acute phase of illness, when IgM agglutinating antibodies predominate, agglutination is easily detected. In this case, there is no problem. This is this phase we have explained, okay? In this phase, IgM antibodies predominate and agglutination is easily detected. Now, after that, IgG antibodies blocking or non-agglutinating antibodies are formed during the course of the infection, and some of them bind with antigens, thus preventing its agglutination with the larger IgM molecules. This is this phase here, okay? In this phase, at this time point, IgG antibodies become blocking. So how to overcome this? So the anti-human globulin is used to bind with the IgG, which is non-agglutinating, and therefore the antigen agglutination can be detected. So that's the idea of this test. The third test is mercaptoethanol test. Now in this test, it's just the same as a standard tube agglutination test, with one difference, that we add something called 2-mercaptoethanol. Now this reagent actually destroys IgM antibodies. Okay, so it breaks down the disulfide bond holding the units of IgM antibody together. So basically it destroys the IgM antibodies. Okay, so any agglutination visible in this test will indicate presence of IgG antibodies, because we destroy the IgM, so agglutination must be due to IgG antibodies. And it will surely indicate chronic infection, because as we have said, agglutination due to IgG is only visible at very high titers of IgG antibodies. The fourth test is complement fixation test. Now this test is used to detect IgG antibodies, okay, and it also indicates chronic infections. Now we're not going to explain this test again. You have studied this test in the virology in first term, so you can go through the picture and you will explain the picture explains everything. The saline diluent is used to dissolve the sample. Just forget about this, not important. Now we move to Yersinia pestis. Now under the microscope, so let's first start with a specimen. So where would I take a specimen to detect Yersinia pestis? Just if you, for your information, Yersinia pestis causes plague, okay? And this plague can be in the skin. It's called bubonic plague, or it can be in the respiratory tract, okay? So it's called pneumonic plague, or it can be in the blood, and in this case it's called septicemic plague, okay? In the bubonic plague, why do they call it bubonic plague? Because there is basically something called buboes. Now the buboes are enlarged subcutaneous nodules. They originate due to Yersinia pestis causing inflammation in the subcutaneous lymph nodes. Okay, so where would I take the sample? Well, if it's a septicemic plague, I take a blood sample. If it's a bubonic plague, if it's a pneumonic plague, I take a sputum sample. If it's a bubonic plague, I take an aspirate from the buboes. So I get a syringe and I withdraw some of the fluid inside the buboes to look for Yersinia pestis. Then first I will make staining, okay? So I will stain the bacteria. It has a special staining pattern, okay? It's called bipolar staining, okay? Bipolar staining, if you look here, the poles of the bacteria stain heavily with the violet stain, while the middle of the bacterium it's not stained, or it's very slightly stained, okay? So this pattern is very characteristic to Yersinia pestis, and you can memorize it by this. The Y has two poles, okay? The Y of Yersinia pestis has two poles, so it has bipolar staining, okay? This way you will never forget it. Bipolar staining. Okay, good. Now we move on. Fluorescent antibody staining. Okay, so this is very easy. So what does it mean, fluorescent antibody staining? This is called direct immunofluorescence. Okay, basically you get an antibody, okay, that is fluorescent. It means it emits fluorescence when it binds to its antigen. And basically you take a serum sample from the patient and you add this fluorescent antibody to the serum sample and you look at it under fluorescent microscope. If you see fluorescence, it means that the antibody has bound to its antigen. Okay, and this antigen is specific to Yersinia pestis, so we know that there is a Yersinia pestis infection. Okay, so basically let's draw this. Now you have a fluorescent antibody. This is a fluorescent antibody. Okay, and you have the antigen. This is, for example, the antigen of Yersinia pestis. This is the Yersinia pestis antigen. Okay, if the antigen is present, if it's there, like this case, the antibody will emit fluorescence. Okay, so the antibody will emit fluorescence. How will I see the fluorescence? I need to look at it by fluorescence microscope. Okay, so I will look at it by fluorescence microscope. So fluorescence microscope. Okay, so that's the idea of the test. The third test we can do is culturing on MacConkey's agar. Okay, Yersinia pestis actually grow after 48 hours on MacConkey's agar and produce lactose-negative colonies. I will remind you that the MacConkey's agar only allows gram-negative bacteria to grow, plus it differentiates between lactose fermenting and lactose non-fermenting bacteria. In this case, Yersinia pestis is non-lactose fermenting, so it shows creamy colonies like this, okay, lactose-negative colonies. Okay, but if you remember that lactose-positive colonies, those which cause lactose fermentation, they show up as red colonies or pinkish colonies on MacConkey's agar. Just a reminder. The fourth test is we can take a sample from the buboes. Okay, we can aspirate the fluid in the buboes and then inject it in guinea pigs or white rats. Okay, and they would die within 2 to 5 days. Now let's move to the serological tests. The first serological test, it's called passive hemagglutination using Yersinia pestis F1 antigen. So F1 antigen, this is a specific antigen for Yersinia pestis. Okay, so how does this test work? Basically, I have the F1 antigen. Okay. Now I could basically add this antigen to a serum sample of the patient. Now let's move to the serological test. We will start with passive hemagglutination using Yersinia pestis F1 antigen. Now this antigen is specific for Yersinia pestis. Okay, so this is how the test works. I get some red blood cells. I add my antigen on them. Okay, I get sensitized red blood cells. It means that my antigens, which are represented here by green plus signs, are stuck on the surface of the red blood cell. Now, the sensitized red blood cells, I will mix them with a patient serum sample. Now, if the serum sample contains the antibodies of the patient, the antibodies coming due to the infection, I will see visible agglutination here. Okay. Now, you might ask yourself, why shouldn't I just add the antigen? I mix it directly with the patient serum and I see if the agglutination occurs or not. The problem of this technique is that the agglutination might not be visible. Okay, so this might produce false negative result. I say the patient has no infection, while he actually has infection and has the antibodies for the Yersinia pestis, but it's just the reaction is just not visible for you. So to make this reaction for sure visible, I attach my antigen to surface of red blood cells, okay, like this, and if the antibody is existing, it will cause the bridging between the adjacent red blood cells carrying the antigens on their surfaces and causing thus hemagglutination. The second test is hemagglutination inhibition. So what does it mean, hemagglutination inhibition? Basically, some antigens of Yersinia pestis can cause agglutination of red blood cells. Okay, so I get a standard antigen. This is a standard antigen. This is standard antigens in my lab. I know this is an antigen that agglutinates red blood cells, and this antigen comes from Yersinia pestis. And I add it to some red blood cells. Okay, and I add the serum of the patient. This is from the patient. Okay, and then I might get two results. If the patient has the antibodies for the Yersinia pestis, meaning he is infected, what will happen? The antibodies, basically the affinity between the antigen and the antibody is greater than the affinity between the antigen and the red blood cells. So in this case, I will see no agglutination. So that's called hemagglutination inhibition. But in the other case, in the case that the patient, in case that the serum does not contain antibody for the Yersinia pestis, meaning no infection, the red blood cells will bind to the antigens, okay, causing hemagglutination. So this is how the test works. The third test is ELISA. Actually, very straightforward. I have the antigen, standard antigen for Yersinia pestis. I will coat the ELISA plate with the antigen. Okay, so coating. And then I will add a serum sample of the patient. Okay, so I will add serum. If the serum sample of the patient contains the antibody for Yersinia pestis, so the antibody will bind to the antigen and it will not be washed in the washing steps we usually do in the ELISA. Okay, so this is from patient serum. And then I add a secondary antibody. Okay, this secondary antibody is usually anti-human immunoglobulin. So this antibody is IgG. Okay, so I add an anti-IgG antibody that is either fluorescent or that is enzyme linked, and then I add a substrate for this enzyme, and I see a color change, and so on. Okay, so I add a secondary antibody. This is a secondary antibody that is either fluorescent or enzyme linked or whatever. The most important thing that if the primary antibody exists, then the secondary antibody will give me a signal that from this signal I will know whether there is an infection or not. Now let's move to Pastorella maltosida. Now usually Pastorella species, usually they live in animals like cats or dogs, and basically the human is infected by animal bites. Okay. Now this Pastorella maltosida causes respiratory tract infection. So basically the specimen I could take is a sputum specimen, and here tells you early morning sputum. It's better always to take a sputum sample early in the morning. And why is this? Early in the morning, you know that during overnight, the cough reflex is suppressed. Okay, so there is no coughing. Okay, and also early in the morning before eating or before washing the mouth. Okay, why is this? Because we want a concentrated specimen in which the bacterial count is sufficiently high so that we can easily detect it. Okay, so that's the meaning of early morning sputum. The second thing, we can take a nasal swab. Okay, and in case of we can also take a specimen from the animal bites, and also we can take a specimen from the cerebrospinal fluid and blood. Okay, then after taking the specimen, we will grow the Pastorella on media containing blood. As we have said before in Brucella was fastidious. Also Pastorella is fastidious. Okay, so as we have said, it needs blood. It needs special nutrients present in the blood. Okay, so we will grow it on blood agar, or basically any media containing blood. Okay, they have a characteristic, the colonies have characteristic color, which is brownish. Okay, and cause no hemolysis. They cause no hemolysis. Okay, and when we grow this Pasteurella on McConkey's agar, they don't grow basically. Okay, so they don't grow on bile containing media such as McConkey's agar. Now we move to a bacteria called Bacteroides fragilis. And now this bacterium is an anaerobic bacterium. It means it doesn't like oxygen. Okay, it lives in anaerobic condition. Now basically it lives as a normal flora in the intestine. So normal flora in the colon basically. Okay, so it lives in colon. Now, infections due to this bacteria occur basically due to injury in the colon. Okay, so due to injury, it moves to the bloodstream. When it moves to blood, it causes infection. Now, so basically now we have understood how the infection occurs. So it stays in the blood mainly. So we will take a specimen from the blood. Okay, so but we need to first decontaminate the skin area before taking a blood sample very well by 70% ethyl or isopropyl alcohol and the tincture iodine. Okay, and then we obtain the specimen. Now, after obtaining the blood sample, you are going to cultivate it using the BACTEC system. Okay. Now, I will not talk much about the BACTEC system, but I have attached a link in the description describing very well the BACTEC system. It's a very nice video. I need you to watch it, and of course, I am sure you will have fun. Okay. Now, this bacteria causes some form of abscess, okay, in the skin too, okay. So this abscess basically contains pus, and we can examine this abscess for presence of pus, and we can also examine for presence of pungent odor, because usually anaerobic bacteria produce very pungent odor, okay. Good. Now, after cultivation in the BACTEC system, let's return to the BACTEC system. After cultivation in the BACTEC system, from the cultures in which there was a growth observed, we will take a sample from these cultures, and then we will cultivate on selective culture media, okay? So the BACTEC system will tell me if there is a growth in the culture medium bottle or not. Now, if there is a growth, I'm not sure whether or not it's Bacteroides fragilis. Okay, so I need to cultivate on selective culture media for Bacteroides fragilis. Okay, these selective culture media, of course, they are anaerobic. And why are they selective? Because basically they contain antibiotics for which the Bacteroides fragilis is resistant, but other bacteria, such as other facultative anaerobic bacteria living in the intestines, are sensitive to these antibiotics. Okay, so these antibiotics will kill the contaminants, like the enteric facultative anaerobic bacteria, but it will not affect the Bacteroides fragilis. Examples for these media is PEA, blood agar. They call it anaerobic phenyl ethyl alcohol blood agar. The phenyl ethyl alcohol, this is the antibiotic, okay? And also we have the kanamycin, vancomycin blood agar. Kanamycin and vancomycin are antibiotics for which Bacteroides fragilis is resistant. Okay, and whatever the medium I will use, I need to put it in anaerobic condition. So how will I will achieve? Now we can achieve the anaerobic condition by two ways. The first way is we get a bag like this. This bag contains some reagents in powdered form. I will add some water to this bag, okay? The water will initiate a reaction, okay? The reaction is a hydrogen. Hydrogen will be produced after adding water, okay? Hydrogen will bind to oxygen inside the jar. Okay, in presence of something called platinum catalyst. The platinum catalyst is present just here. Okay, this is the platinum catalyst. And the result of this reaction is water. So basically we are turning oxygen into water. Okay, so after some time, the medium, the air inside after some time, the medium inside the jar will be anaerobic. Good. Okay, so how will I know that the medium has successfully become anaerobic? Basically, there is, if you can see here, there is a blue color on the bag. Okay, this is a methylene blue indicator. Okay, when the medium becomes anaerobic, it turns white. Okay, its color turns white. So its color is white when no oxygen remains in the jar. But if there is still oxygen, okay, it's blue in color. Okay, so we are still in the first way of making the medium anaerobic. Okay, so we put a bag. We have a bag like this. It contains powdered reagents. I add water to this bag. The water initiates a reaction that results in the production of hydrogen. Okay, the hydrogen interacts with oxygen in the presence of a catalyst. The catalyst is present just here, and this reaction will produce water. So we are turning oxygen to water. Okay, so this catalyst is palladium-coated aluminum pellets. Sometimes they use palladium, sometimes they use platinum, and an indicator. Okay, so this is the first way. Okay, so this is the first way of making the medium anaerobic. The second way is basically I will not use this bag. I will use something called oxide jar. Okay, as you can see in the oxide jar, there are vents here, okay, one vent here for introducing the gas, and one vent from the other side for extracting the gas. So first of all, I will attach I will attach a vacuum. Okay, the vacuum will remove the oxygen. Okay, will remove the air inside the jar. And then I will attach a pump here. The pump will introduce a gas inside the jar. This gas is composed of 85% nitrogen, 10% hydrogen, and 5% carbon dioxide. This is basically an anaerobic condition. Okay, this gas composition is anaerobic. After that, we will incubate. So here it tells you that you have to place the plates in a medium containing the same composition of air, the same composition, okay, as this composition before cultivating the bacteria on it. Okay, it's like a conditioning. It's like you are conditioning your plates before cultivation on it. Okay. Okay, after incubation, of course, colonies will grow. Okay, and you will examine the shape of the colonies. You will make gram stain. Okay, you will make biochemical tests, enzyme assays, and GLC. Okay, GLC is used to detect unique short chain fatty acids that anaerobes produce. Now, you don't need to memorize these percentages. Okay, just read them. Don't memorize them. We will not ask you what are the percentage of blah blah blah. Okay. Okay. So Legionella. Legionella is a bacteria that usually loves to stay in water. So basically, it's present in water that is not moving. Okay, like in lakes. and it's also present in air conditioners that are not cleaned regularly. It causes two conditions, the Legionnaire's disease and Pontiac fever. Okay. Now, Legionnaire's disease is a form of pneumonia, while Pontiac fever is just fever. Pontiac fever is a milder disease that goes away by on its own. Okay. For the names Legionnaires, it comes from there was like a meeting between American war veterans. So it's called American Legion Convention. So this meeting resulted in several cases of the people attending the meeting having pneumonia, and when they isolated the bacteria, they found it was Legionella. So that's why they called it Legionnaire's disease. And Pontiac comes from, basically, it's the name of a district in Michigan, USA. There was also an outbreak of this fever in that particular place, in that particular district. That's why they named the name of the fever after the name of the district. But however, the origin of the names is not important. It's just for fun, just for you. When you study it, you just you just don't find the names weird. Okay, so you become familiar with the names. Now, for the diagnosis of Legionnaire's disease, now these two conditions basically are different concerning the symptoms they cause, but they are very similar in diagnosis. Okay, so first, Legionnaire's disease. There are three conditions that makes us suspect that there is Legionnaire's disease. First of all, the polymorphonuclear leukocytes in the sputum, but no organisms, few or no organisms stained by Gram stain, because these organisms are intracellular. Legionella is an intracellular organism that is stained poorly by Gram stain. This is number one. Number two, hyponatremia. Number three is high temperature. So, as we have said, Legionella is an intracellular pathogen not stained well by Gram stain. So then there is another staining technique called Gemini's method. Just know the name, okay? And you can connect Gionella to Gemini's, like they both have G's in them, okay? But the details of this technique are not needed from you, okay? The second test we can do is direct fluorescent antibody technique. Okay, it's very straightforward. Direct fluorescence, indirect immunofluorescence. We have an antibody. We have actually explained this before. We have an antibody attached to a fluorescent probe. Okay, and this antibody is specific for an antigen, a certain antigen on the surface of Legionella bacteria, and so basically, if we add the antibody to the specimen and observe using a fluorescence microscope, if we find this green fluorescence like this, then we know that there is an infection with Legionella. Okay, good. Now, here he tells you, when you take the specimen, you treat the specimen with acid, especially the sputum specimen, because you need to kill the contaminating normal flora present with Legionella. So Legionella is resistant to acid. So you add acid to the sputum sample to kill the contaminating normal flora. Then you culture the sample on CYE agar. It's short for charcoal yeast extract agar. So charcoal, basically it functions as an inhibitor of toxins. Okay, so your Legionella, when it grows, it produces toxins that actually, when they accumulate, they might inhibit its growth. So the charcoal functions to inhibit the activity of these toxins. And yeast extract is just a nutrient that the bacteria uses to grow. It contains L-cysteine. This is another nutrient. antibiotics to make the medium selective, and dyes. The dyes actually, when Legionella pneumophila grows due to these dyes, it gives blue-green colonies, as we can see here. Okay, the greenish color is not very evident, but the blue color is more evident. Now, serological testing. So the first test we took, we can make Geminis stain and direct fluorescent antibody staining. Okay. And we can cultivate on CYE, charcoal yeast extract. And then serological testing. Okay, we can apply serological testing to a serum sample obtained from the patient. The serum sample can be obtained in the acute phase. Acute phase, it means after 1 week of onset of symptoms. Okay, so 1 week after onset. Of symptoms. Okay, a convalescent serum sample, it means 3 to 6 weeks after onset. 3 to 6 weeks. Why we need 2 samples? Because we need to monitor the antibody titer. Okay, so we would monitor the IgG antibody titer, and if there is a 4-fold increase from the acute to the convalescent sample, if there is a 4-fold difference if there is a 4-fold rise in antibody titer in the convalescent serum sample compared to the acute serum sample, then this confirms the diagnosis. And here they use an immunofluorescence antibody staining technique. So we previously determined the antibody titer in the Brucella, if you remember, using the dilution technique, using agglutination. Okay, using agglutination technique. Here we use another technique to determine the titer using immunofluorescence. Okay, so basically the idea is simple, but these details are not needed from you. They are just for you to because nothing seems weird to you. We will take the serum sample. We will make serial dilutions just like in Brucella. But in this case, we will use each serum dilution, we will use it. We will use each serum dilution with an antigen, with a standard antigen, okay? And the highest dilution showing fluorescence will be considered as the antibody titer. In the Brucella, the highest dilution showing agglutination was considered antibody titer. Now we move on to urine. Actually, the antigens of the Legionella live for a few days within the urine and can remain actually. So the antigen of Legionella can stay in the urine for several weeks. Okay, starting from few days of illness after onset, and can remain for several weeks. Okay, so it's a very reliable technique to detect Legionella because it stays for long. So I will detect the antigen using radioimmunoassay or ELISA. Pontiac fever is diagnosed also serologically. Thank you, Lucas. I hope you study hard and be safe. Stay safe.